Good evening, and I am excited that everyone is attending our first AASH 1-hour webinar on virology, the study of viruses with Dr. James Meegan. He is the husband of Rhonda Raffi Meegan, our AASH Vice President. So welcome, and we're so glad that you can be with us tonight. It will be very interesting and quite a learning experience for all of us and get us up to date on this topic. First of all, Rhonda Meegan will start out with a coronavirus prayer. Rhonda?
Lord Jesus, you heal the sick. We pray that the coronavirus that has affected so many in our world can be contained, controlled, and cured. We lift up to your mercy all those affected, all those working hard to study and cure this illness, and all healthcare professionals and emergency management teams that are working day and night to keep us safe. Help us work to trust in your merciful care. Lord Jesus, hear our prayer. Amen.
Amen.
Okay, now I'll turn it back over to Deb.
Thank you, Rhonda. First, I want to give you an introduction on Dr. Meegan. He is the former Director, Office of Global Research, National Institute of Allergy and Infectious Diseases of the National Institute of Health. He worked under Dr. Anthony Fauci, of whom we're all very familiar every day on television. His research programs have focused on the role of viral diseases in international health and tropical medicine. And James Meegan received his PhD from the University of Connecticut and has had a long career in science, specializing in microbiology, virology, infectious diseases, and arthropod-borne and hemorrhagic fever viruses. He has held research positions at the University of California at Berkeley, Yale Medical School, United States Army Institute of Infectious Diseases, and He has held leadership positions at the World Health Organization in Geneva in 1988 to 1992, the National Institutes of Allergy and Infectious Diseases at the United States National Institutes of Health, and was a Senior Director of Research and Development at Vitrogen Life Technologies Corporation. Jim has had many distinguished awards, published numerous scholarly manuscripts, and has been a consultant and advisor to various individual countries, scientific groups, and commissions. Jim, thank you very much for taking time to share all of your knowledge with us, well, at least a small piece of it, and for being here this evening with all of us. So go ahead, we're glad to have you here. Thank you.
Well, thank you for inviting me. I've got to start with a few disclaimers. I'm speaking for myself tonight, not for any organization or any government. And additionally, I'm not a clinician, I'm a researcher. So any questions related to your health should be directed to your own healthcare provider. I'll go through a few quick introductory comments so that we're all on the same page. I've looked over all the questions that have been sent in, and I want to cover a few of the common themes. And then Deborah will help with some questions that have been coming in since. So this unfortunate person here, Dr. Cockburn, in 1963, he's unfortunate because everybody who works with infectious disease usually shows this quote where he thought that infectious diseases were, in essence, dead. He had pretty good reason for this. As you see, we were doing a good job, especially with the introduction of antibiotics, to controlling infectious diseases. I'll call your attention to one thing here, the 1917 spike, the Spanish flu that we'll talk about a little bit later. But the story really wasn't that we conquered infectious diseases. In fact, it's more of a perpetual challenge. And we've known this, not only Dave Morens and Greg Folkers and Tony Fauci in this article, but we've known it before then. Diseases are going to pop up all the time, some of them re-emerging and some of them brand new, and some of them might be quite challenging. Here are 20 of the worst epidemics and pandemics in history, and as you can see, they date back to 3000 BC, and they cover a lot of different geographic areas. They also cover things like polio that are transmitted by fecal-oral route, flu epidemics that are respiratory, yellow fever that's mosquito-borne. So our history has been a series of attacks by pathogens. We sometimes think of man's predators as something like a great white shark, but in essence, these small pathogens, microorganisms, are really our predators. And as you can see here, in many of these larger epidemics, significant number of people died. Call your attention in particular to the last two, the 1918-19 Spanish flu with over 50 million, and HIV, which has killed over 25 million. Indeed, if you go through many parts of Europe, shown here is Austria on the left and the Czech Republic on the right, you'll see monuments to plagues, to the recovery from plagues. And it was just a series over and over and over again of different plagues. Here's a slide that I started with Dr. Fauci way back when, last probably 4 decades. You don't have to read all of it. It just shows that even recently, we've had lots and lots of emerging and re-emerging diseases. You can see Ebola mentioned there. You can see the different influenzas, SARS, a number of different organisms, including antibiotic-resistant bacteria. Like I said, it wasn't unpredicted. The World Health Organization each year puts out a list of the biggest threats to global health, and this is part of that list. And as you can see, under global influenza pandemic, they were concerned that the world will face another influenza pandemic or similar disease. And so people have been worried for a long time about emerging diseases. This is a good example of one that occurred in the United States. West Nile virus came in 1999 and since then has established itself. It's a mosquito-borne disease. It's a good example of how diseases can emerge. Influenza, we see almost every year, and some years there's a large change and it causes pandemics. Certainly the influenza pandemic of 1918, with its large number of deaths, well over 500,000 in the United States, has been compared to the current COVID outbreak. And it's very interesting, in that year, the green line is what usually happens every year with influenza. Young people and older age groups are the ones that die. But in that year, not only those groups, but young people died. That was the big difference that occurred with Spanish flu. And if you look at some of these pictures, they're very reminiscent of what we're seeing today, having to set up convention centers in some states in the United States where they're putting in extra beds. All sorts of masks with school children or athletes and people bringing in various patients to be treated. Churches, schools, everything closed down. Transportation, and as you can see down here in the bottom, everyone must wear flu masks. So the parallels are haunting. Interestingly enough, in those days, like today, they didn't have many things that they could use to prevent flu. And here are some of the things that they recommended. Sort of interesting to see, keeping your feet and clothing dry and avoiding crowds. Some of them are very similar to what we're talking about today. Why do these diseases emerge and reemerge? A lot of it is based upon human behavior, some of it on technology, some of it on breakdown of public health measures. Not vaccinating is a good example, with measles coming back. Changes in the ecology and the environment. Certainly with COVID, people crammed together with animals. High density is one of the major concerns. Another has been, of course, the spread of mosquitoes. Mosquitoes have come back in a big way in many of the developing countries and have led to the diseases like Zika. But there's plenty of ways that we interact with animals and can transmit viruses back and forth. And this is probably exactly what happened with COVID, a bat being the potential reservoir. You've probably all seen this picture on the left of the coronavirus. Looks a little bit like a soccer ball with lollipops sticking out of it. And in fact, it was a shame coronavirus. I have heard that coronavirus, Corona Beer has actually decided to change their name to something a little less pathogenic. These are the symptoms that I think everyone is familiar with now. Certainly shortness of breath and loss of taste and smell seem to be highly characteristic of this coronavirus. We've also known that a number of the people who are older in age or immune compromised or obese or have heart conditions are at a high risk. And this has been said over and over and over again. Methods, the only tools we really have, the only public health tools that we have right now until we get a vaccine to help us prevent this disease. And we'll be talking about some of these things in the question and answer period in a little bit more detail. Thought I'd include this since we're coming up on the flu season, and I got my flu shot last week. It's certainly a concern of everyone that an already overburdened healthcare system will then be further overburdened with the flu season. So if we can prevent flus from occurring with a vaccine, that's why everyone's recommending it. You'll see a little bit of differences between COVID, flu, and allergies. Allergies is a lot of sniffing, sneezing, runny noses, itchy eyes. That's sort of characteristic for the allergies. Where COVID, that shortness of breath and the loss of smell and taste are certainly some of the things that can help you separate these three common problems that we're facing as we go into the colder months. Unfortunately, as you know, both in Europe and in the United States, we seem to be entering the second wave. And it appears like the virus is winning in many cases. So a lot of emphasis has been placed on the need for the vaccines, and certainly vaccines have helped us for years and years and years. A number of diseases have been controlled by vaccination, and of course, billions and billions of dollars are saved by not having to take care of children with measles or polio. And we've had success here. You could see in the bars. This is tetanus. It's almost non-existent now with the advent of the vaccine, and with measles as well, except for a resurgence we've seen, but nothing like the cases that we saw in the 50s and 60s. So where do we stand on vaccines for coronaviruses? You've heard people discuss phase 1, 2, and 3 as clinical trials. This gives you an idea of where the vaccines stand. 11 of them are in advanced testing, and that's very good. As you know, there are a number of different methods, and vaccines often fail at the very last phase. And so it's good that we have 11 possibilities here. Now, these are made in methods that were used many years ago, such as the ones listed here. Things like yellow fever, they took the yellow fever virus and weakened it, and that's the vaccine. It's called live attenuated. Others, they took the virus and killed it with formalin, and that's a kill vaccine. And in others, usually bacteria, they break it apart into subcellular components. Those are the three standard old-time methods. But now we've got all sorts of new methods that you've probably heard about. Naked DNA vaccines, subunit vaccines, all sorts of virus particles, edible vaccines. And so I want to just go over a couple of those that you've probably heard of. The leading candidates include some of these new vaccines. So again, here's a little bit of a cartoon, but it's pretty good, talking about which parts of the virus we really want to make a vaccine against. And if you look at the surface of this, the E protein, which is mentioned up here, is a protein that once the virus attaches to a human cell, it's like a can opener, and it lets the virus open up, get into the cell. Over on the right, the large brown lollipop, if you will, is what grabs onto cells. So as the virus tumbles through your respiratory system, these little spikes are what attach to the cells, and then that E protein opens up the cell and it goes in. So what we're trying to make vaccines to are these two proteins, focusing on the lollipops, the brown external spikes. If we could make a vaccine that blocks those, then the virus can't infect your cells. One of the ones that you've probably heard a lot about is the Moderna vaccine. It's also a vaccine used by the Oxford group in England. And it's called a messenger RNA vaccine. So instead of taking the virus and killing it or weakening it, what they do is take the virus and take out of it the instructions that the virus has in its nucleic acid, in this case, RNA. And they take that RNA and put it into a little capsule of lipid, of fat, and they inoculate it into your arm, and your body, your muscle cells, absorbs that, and the virus gets into your own cells, and that message starts making the little lollipop of the virus and dumps it out into your circulatory system. So your own body is sort of the manufacturing plant, and all they do is they send in the instructions. And this is a new type of vaccine. It's the one that's leading the way because it's easy to make. You can make a large number of doses. There are some challenges with it because the vaccine itself has to be kept cold. But this is one of the new ones and probably is leading the way right now. But it is a new method. There aren't any vaccines like this on the market right now. So it will be challenging to show not only that it works, but that this method is safe. A second one that you've probably heard of, this is the Russian vaccine. As you know, both the Chinese and the Russian have made and are selling vaccine. Instead, they did phase 1 trials, phase 2 trials, and they skipped phase 3 and just started selling their vaccine. Phase 1 trial is safety in maybe less than 100 people. Phase 2 is further safety in 1,000. Phase 3 is to look for efficacy in tens of thousands. They just skipped that. And this adenovirus, this vaccine that we're going to talk about now, is the one that's being sold by the Russians. What they do is they take the gene for the coronavirus and put it inside an already existing vaccine. And that vaccine then carries it in to the person like a Trojan horse. It's a new methodology as well. And so it will have to be shown to be safe and effective, just like the mRNA vaccine. So those are the two that are leading the way now. There are other methods. There is a live attenuated, there is an inactivated vaccine, there are subunit vaccines. So there are a number of them. And down on the bottom line here, you can see some of the companies that are involved with all of these different methods. And I think one of the questions we have asks, which of these methods is going to be good, and do you have to mix and match? And so we'll talk about that in a little while. So in the long run, Josh Lederberg, this is a quote from Josh Lederberg, a Nobel laureate, we're really fighting these pathogens for the rest of our lives, for the rest of humanity. When we think of evolution, we think of man and how long it takes even to change our genome a little bit. But you have to remember that these viruses and these bacteria replicate overnight so quickly into large numbers of generations that they can change their genome overnight. So they're always going to have that advantage over us. We might develop a vaccine. They might develop a way to get around that vaccine. They have this permanent advantage of being able to replicate so much faster. So I think we should go into some of the questions and answers. I think you have those, Deborah?
I do, yes. Thank you, Jim. And just want to remind you that to go ahead and put any questions that you have into the chat screen down below on the bottom of the screen there, and we will try to get to all of those that we can. And so, Jim, one of the first questions is from Marcia. Do you think this type of pandemic can and will happen again in our lifetime?
You just talked a little bit about this in this question. Yes, I think these pathogens are going to be evolving quickly. And as we've shown, even within the last few decades, there have been a lot of these. So we all thought that the most likely pandemic was going to be another influenza. Turned out to be a coronavirus. Are there others out there that could do this? Perhaps. So we're going to have to be vigilant. Yes, I'm afraid we're going to be fighting these pathogens and our children will be fighting them forever.
And what are your predictions for the timing of a vaccine?
Okay, there's a little bit of a semantic argument there. If you have a vaccine that looks like it's working at the end of the phase 3 trial, again, with tens of thousands of people, some of the vaccines are in about 30,000 people in multiple countries now. The minute you show it's efficacious and safe, you have a vaccine, but you don't have it manufactured in large enough amounts to give. If you think of the United States, we have 350 million people. If you needed 2 shots, that's 700,000 or 700 million syringes. I'm not sure we even have that many. The logistics of delivering that vaccine have to be included. So yes, you might have a vaccine soon, probably at the end of November and December, if we're lucky. But you won't have it available until April or May to everyone. And with limited supplies, there'll be some ethical questions as to who gets it first. Do the healthcare workers get it first? I think most people would say that. But then do old people? Or do children get it? Or does the military get it? All of those questions will have to be answered.
Okay, thank you. What was the source of this virus?
Okay, there's been some question about that, but I think it's been answered by the molecular virologists. They've looked very carefully at the sequence of this particular virus. If you were to try to make a virus like this, you would take an existing coronavirus that you knew infected people, and you would add into it molecularly pathogenic genes to make it more dangerous. There's no evidence of that being done. Not only is there no connection with a previously existing coronavirus, there is some, but not an enormous amount. And there's no telltale signs of putting in a gene. When you put a gene into a nucleic acid and alter it, there's always two ends that have to be the exact same. There's no evidence of that. So looking at it very carefully, it probably came from nature, spilling over from an animal into a human. And now that doesn't mean that it couldn't have come out of the lab in Wuhan. I've worked in the lab in Wuhan, actually. It's a good lab. Like everywhere in the world, there are young virologists going out and getting samples from rabbits and making a thesis on the viruses in rabbits or the viruses in mice. And there are probably people going out and getting samples from bats. It's possible that they brought it into the lab and it somehow got out of the lab. Unlikely, but there's no evidence that it was a bioweapon of any kind or altered in any way.
Okay. Is plasma antibody treatment effective, and did it help the president?
So what we're trying to do with vaccines is to make your body make antibodies. What we can do is take someone who has recovered from the disease, who has a lot of antibodies, and collect their blood and isolate the antibodies that they have. We used to do this many, many years ago. Many of you might remember that when we traveled, we'd get a shot of gamma globulin. That's exactly what they did. We didn't have hepatitis vaccines. So they would get a large pool of people's serum and isolate all the antibodies and hope that they had people in that pool that had anti-hepatitis, and they'd give you a shot before you went overseas. So we've been looking at this sort of methodology for a long time. So you would take a patient and give them antibodies so that they could start fighting the virus right away. We'd give them antibodies that would attach to that little lollipop that we were talking about earlier. The key to this is to get it early on in the disease. In fact, the key to any virus infection is to treat it as early as possible. So it probably will help, but it's probably highly dependent upon how quickly the hospital or the doctor sees you and can get that material into you. With the president, he was being tested every day, and he got the material as fast as anybody in the world got it. So it was the optimum situation for him, and perhaps helped him quite a bit. Most people wouldn't be tested every day. They'd go in when they didn't feel, when they felt sick. They might have to wait 2 days for the test to come back. So you're further along in the disease, it might not help you, but it's a good possibility, and it's being tested now and researched now. And again, it wouldn't be unusual to see that you really have to give it early, that it doesn't help you later on. But there are a number of other medicines now that are based upon antibody therapy for other diseases. So it's a good thing to be studying.
Okay, and question from Stephanie. With so many vaccines and different chemistries being developed, are people going to need to vaccinate themselves several times, and perhaps with different vaccines made by different methods?
That's a really perceptive question, Stephanie, in that we don't know with these different methods that we were just talking about, whether one will produce an antibody profile that's more protective than another one. So we really don't know the answers to that yet. As we go through and test for efficacy, we'll know whether one vaccine with 2 shots is going to be effective, or another vaccine with 2 shots or 1 shot will be effective. But we won't know whether it would be best to take a live attenuated vaccine from Pfizer and then go and take a messenger RNA vaccine a month later from Moderna, and that you'd get a broader response to be more protective. We probably won't know that, but we will know whether one vaccine with 1 or 2 shots will be effective.
Okay. And Another question is, to whom should we listen? And from whom should we take our cues for COVID-19 matters?
Well, as you know, the internet is filled with virologists all of a sudden, experts all over the place. I think you should stick with the scientists. There's been some political aspects that have been sneaking into these arguments too. So, you know, I used to work at NIH. I retired in September. It's sort of funny. I was hoping that after I retired, I wouldn't be seeing Dr. Fauci every day, but now I'm seeing him every day on TV. So I think he's a non-political person. He's worked for 6 or 8 different presidents, and he's a scientific nerd, if you will, and I'd follow the scientists and take their advice. But the internet is like the Wild West. It's filled with all sorts of crazy things.
Wild West, yes. What are the differences between the tests that are being offered, the PCR, the rapid, and the antibody tests?
Good question again. So the PCR test, that takes a little bit longer, is a test looking at whether that sample from your nose or your mouth has virus in it. It's a very, very, very sensitive test, but it takes a few hours, probably 3 or 4. So it's not considered rapid. But it gives you, right then and there, tells you that moment, is that person, does he have or she have virus? Recognize that's only a snapshot. The next day, that person might get exposed and get the disease. So if you tested negative 3 days ago, it doesn't mean you're still negative. You might have got it exposed. But it's very sensitive. When you're making these diagnostic tests, if you want to get them faster, you usually have to give up sensitivity. And that's what the rapid test is. It's similar to a pregnancy test on a little filter paper, but it's probably much less sensitive, 100-fold less sensitive than the PCR test. But it's rapid. You can do it in less than an hour. And that's the test that was being used in the White House and many other places because it's so convenient, but it's not as sensitive. It again is looking for the virus. In that case, it's looking for the protein part of the virus. The PCR, polymerase chain reaction, was looking for the nucleic part of the virus. So that test is quick, but a positive tells you you're positive, a negative doesn't always tell you you're negative. You might miss some. The third test isn't looking at whether the virus is in you now. It's an antibody test. If you go through the disease after 10 or 12 days, your system responds with antibody, starts controlling the virus, and the virus disappears, and you develop antibodies. So from about day 12 or 14 on, you can take blood and look for those antibodies. And that's oftentimes called an ELISA test. And that's looking at antibodies. It tells you that you used to have the disease, that you recovered from it. And there are 2 types of antibodies you can look at. IgM that come up quickly and go down within a couple of months, and IgG that go up and stay. And so that test is telling you not whether you have the virus now, it's telling you whether you had it a few weeks ago or a few months ago. So those are the difference. 2 tests to look for the antigen in you right away, and 1 test to look at the antibody that's a marker that you had been infected.
Okay. And what is herd immunity?
And that's mentioned a lot. What it means is that roughly 70 to 80% of the population have antibody and cannot transmit the virus. So let's say, use measles as an example. If we vaccinate most of the people in the United States, and then we will have herd immunity from the vaccine, many of those people will have antibodies and be protected. And the virus can't circulate because the virus can't find a host, a human, that isn't protected, so it can't transmit. So the question is, how high do you have to get your antibodies to get herd immunity? And it's roughly 70 or 80% with other respiratory diseases. So Sweden, as you know, decided not to do an aggressive campaign against coronavirus and instead tried for herd immunity. They don't have herd immunity yet, and their death rates per 100,000 are more than France. So it didn't work for them. So to get to herd immunity without a vaccine, we'd have to let 80% of the people get infected. We've seen so far, we've infected about 10% of the population and have 200,000 people dead. Are we willing to infect another 10 and 10 and 10 and 10 and accept another 200, 200, 200? I don't think that's going to work.
Okay, thank you. Rhonda's going to go ahead and do the questions that came online, please.
One more. Okay.
So thank you, Jim.
Hi, I've got my AASH mask. Jim, there was a question. What about masks?
Okay, this is a big question that's come back and forth. I think the scientific data is pretty clear. Masks protect you somewhat and protect you from infecting other people. There are two types of masks. There is a cloth mask that Rhonda's demonstrating for you right now. And there's also an N95 mask. It's actually a HEPA filter mask. That mask, if you put it on, it's hard to breathe through, but it will filter out all the viruses. That will protect you. This mask won't do that, but it will protect you from large droplets. So people will say, look, the virus is so small it can go right through that mask. And that's true, but most viruses are shed in droplets from other people. And those droplets are big. In fact, an individual virus coming out of a person's nose will probably be inactivated pretty quickly because it doesn't react well to low humidity and, temperature changes. So with a mask like that, you are getting protection from large droplets that can't go through. You're also, if you're sneezing, and we know that a lot of people don't even know that they're infected, you're also saving other people. Excuse me. So you're doing a good thing for humanity because you're not allowing your droplets to go out into the supermarket and infect someone else. So it's pretty clear to me as a scientist that masks are worth wearing. We saw in some of the pictures, it was used effectively against influenza. I'm an advocate of wearing masks as much as you possibly can.
Okay, Jim, I've got one from the UK, from Roxana from UK. What would happen if those least likely to suffer from the virus, for example, everyone under 45 years of age, were given the choice of participating freely in community life with no isolation, and all those likely to suffer serious health consequences or death were told to isolate as much as possible? Would this achieve the so-called herd immunity?
That's a good question. The problem with that is that we don't know about long-term consequences of this disease. So when we say that a person has a minor illness, their lungs are affected. And I'm worried that in the long run, many of these lungs are going to be scarred, even though the person had a relatively mild disease. And we've seen some scientific data of that. If you've talked to people who recovered as well, many of them have what is being called long hauler symptoms. They've lost their smell, they've lost their taste for months and months. I know two people in particular who cannot smell anymore because of this. We've seen with some of the athletes in the United States, myocarditis. Their heart is actually swollen because of this, even though they don't have significant disease. So the problem is we don't know whether there are long-term problems even when you have a minor disease. And those people would be shedding virus into the population where there might be an immune compromised person. For example, think about your child in school in an average class of 20 or 30. How many other kids are on inhalers because of asthma? Probably 2 or 3 in every class of 20 or 30. So, and many people don't know that they have an underlying disease. So there are significant problems with that methodology. And again, many people have heart disease and don't know it. Many people are obese. Certainly in the United States, we have an epidemic of obesity. Are those people going to get a mild disease? We just don't know. So I don't think that methodology is going to work.
From Rosalie, do we know why the virus has such different effect on different people?
No, we've got a lot to learn about this virus. Certainly, one of the things that was interesting was that the virus causes small coagulations in vessels in your body. And we didn't know anything about the pathogenicity of this disease. Again, it's a new disease. But it seems to be multi-organ. So some people get only lung disease, some people get cardiac problems as well as lung disease. Some people have encephalitis or an effect on the brain as well. So we don't know much about it. We're learning a lot about it now, but we don't know much about the pathogenicity, and we don't know why some people have one course of the disease and others don't. There's been some papers out, and it'll have to be studied more and more, that some people with different blood types are more susceptible. So there's going to be a real gamut of different diseases, a whole spectrum that we're going to learn about. And maybe the host, maybe the human's underlying conditions will dictate which way the virus will affect them.
And from Brooke, aren't there 2 vaccines that have been halted? Why exactly?
Okay, a lot of people are concerned about that. This is normal. It happens all the time when you're looking at vaccines. Remember, these are the phase 3 trials. You take 30,000 people, you divide them in half, you give half of them your vaccine, the other half you give a placebo, just salt water. And then you let these people out into the world and you're hoping that they get exposed. After a period of time, 1, 2, 3, 4 months, you look at how many of them that had the vaccine got sick and died versus how many who got no vaccine got sick and died. And hopefully you can show the efficacy in those trials. But everyone is blinded in these trials. You've heard that term. Nobody except one independent committee that oversees the trial. Nobody knows whether you got the vaccine or not. So let's say you have a car accident and die. Oftentimes they'll stop the trial, even though they don't know whether that person got the vaccine or the placebo, and even though it doesn't seem to be related. And then they'll do a quick review. This independent clinical group will come in and say, did the vaccine possibly cause this person to have some sort of a seizure that caused the accident? So every unusual event is reviewed. And we really don't know whether these trials were stopped for the vaccine or for the placebo group. So it's not unusual. It's good safety. An independent group comes in and says, we're going to look at this and see. And if they will then say, let's see if it's in the placebo group or not. Let's see if it's normal or not. So there are 2 trials that are halted. I hope they can get back on. It's not unusual. Let's hope that they're not serious. But it's telling us that we're following good safety to stop these trials and to look at every single event.
And anonymous, how likely is someone to have a second case of the virus?
Another very good question. There have been a small number, less than 25 cases, documented cases of people being reinfected. Now, think about that number. That's a very small number when you consider the millions of people who have been affected. So if we focus on that and say, well, you can get reinfected, we have to also consider the fact that a lot of people are not getting reinfected. There have been at least 2 cases that have been well studied within those 25. In one case, they got a minor disease. In another case, they got a more significant disease. So the question is COVID like other viruses that when you recover, you usually have good immunity for many years? Or is it a virus that's unique and when you recover, your immunity disappears quicker? We're worried that it's that latter because other COVIDs, their immunity goes down over a year or 2. That might mean that if we have a vaccine, we've actually got to make the immunity that we develop with a vaccine better than what you would get if you had the disease, and we can do that. So we're watching those reinfections carefully because they're telling us how long the antibody stays in your body and protects you. But it's a small group so far that have been reinfected, and we'll be watching it and studying it as we go along. But it doesn't mean that we can't make a vaccine. When you think of it, you get infected by your respiratory route, but you get vaccinated in your arm by a needle. So we actually have, in many cases, vaccines that induce more antibody than if you had gotten the disease and recovered. So there's hope still that we can make a good vaccine to coronavirus.
Okay, from in Brescia, IgG versus IgM. Is there enough data on either for the data to be definitive? And how much time would you project for that data to be definitive?
What you need to do is ideally to bleed someone every day for a year and look at their antibody comes up and look at the IgG and the IgM. I laugh because the person that we could do that with is probably the president, who's getting bled every day, and his wife. And so they would be interesting to study because we've got bloods and samples from them on every single day. So without that, you have to sort of bleed various people and say, how long were you infected? Okay, 2 weeks. Okay, you were 4 weeks. Okay, and then in each of those samples, test them for IgG and IgM. With other viruses, IgM comes up a little bit before IgG, spikes high, and then disappears to low levels, not totally gone, but very low levels within 6 months. And the IgG stays longer. I would anticipate from the preliminary data on IgM that it will disappear within 2 months with coronavirus. The IgG will stay longer. So let's say you were worried, did I have the disease back in December or January? If you did, they bled you and they found IgG but not IgM, means you had the disease way back in December. If they found IgM and IgG, it probably means you had the disease a month ago. So we don't have the data for coronaviruses. We have the data for many other viruses. I anticipate that IgM will be a valuable tool, and looking at IgG and IgM will be valuable public health tools for us.
Okay, from an anonymous, would it be possible or advisable to take one of each type of vaccine?
I don't know. It's an interesting thought. Again, a perceptive question. We'll have to see when the vaccines come. As I said, each vaccine with these different methodologies that we talked with will induce a little bit different response. So I think what the questioner is saying is, if I got one type of vaccine and another, will I have a very broad response? We probably won't know that for a long time. We'll know about one vaccine and another vaccine, but I doubt you're going to see one company give their vaccine to another company and say, why don't you try them together? So we won't know that. I think what we're going to have to do is look at the data and say, okay, I'm going to get my shot and my booster of this particular vaccine and stay with that, and then watch it from then on. But very interesting question. For most diseases, we don't have more than one vaccine or type of vaccine. They're very similar. So this will be one of the first times that we'll hopefully have 3 or 4 or 5 different types of vaccine.
Okay, Jim, from Nancy McKenzie. What timeline do you believe that the president tested positive for the virus?
I don't know. The question is, was he being bled or tested every day? And we think he was. Then the second question is, were they using the really sensitive PCR test, or were they using just the rapid, less sensitive test? We think it was the rapid test. So we really don't know. Now, they probably kept his samples and could go back and figure it out. But one argument is, gee, the president should have been getting the best test. He should have been getting the PCR. And there's really not a lot of reasons. I could do a PCR for him overnight. Most labs could. Why not? Why didn't he have the most sensitive test? So we probably don't know exactly when he was infected. But if they went back and did the PCR on all his stored samples, they'd be able to determine it pretty well.
Okay, Jim, I think this is overseas. Anonymous, does it get weaker as it is transmitted within a family?
Another good perceptive question. If you look at past epidemics of different viruses, sometimes they get, as they go through the population, they get weaker. Sometimes they get a little more virulent. It brings up the questions of mutations. As it's going through a family, are they collecting mutations that would make it weaker or more lethal? Mutations occur all the time. They're occurring in us, they're occurring in viruses all the time, and most of them mean nothing. They're silent mutations. But every once in a while, one of them might make the virus less virulent or more virulent. So we're watching for mutations. You'll hear them say, oh yes, there's mutations, but oftentimes that doesn't mean anything. The nucleic acid changed, but the virus is still behaving the way it was before. So we don't know whether it will attenuate, that's the word for getting weaker, as it goes from person to person to person. We just don't know that now. And like I said, historically, viruses have gone different ways. There are all sorts of elements that affect that. For example, let's say your disease got much more lethal. The person would go to the hospital quicker and be taken out of the population, and that virus wouldn't spread very quickly. So from a virus's point of view, it's not good to be too lethal. From a virus's point of view, you want the person coughing and walking all around and spreading the virus to everyone. So oftentimes, the virus that does end up circulating is a little weaker. We'll just have to see with it. There's so much, people want definitive answers, and with a new disease, we're just learning as we go along.
Okay, not sure this is COVID related, but someone, I'm sure you can address it, Jim. From the Catholic point of view, is there testing on embryos? And if so, which labs are doing this testing? Are you?
On embryos, no. So the only question that the Catholic Church has in the past has been whether the cell cultures and cell cultures are cells grown in the bottom of a petri dish that the virus infects and multiplies in, whether those had come from an abortion or not. And some of them have. And so that was a concern for I think it was for mumps vaccine. They used cell cultures way back in the old days that had come from the hospital that were fetuses, and made cell cultures out of them and then produced the vaccine in it. So there is some concern that is that vaccine tainted somehow? No one's doing any studies on human embryos. The only thing they could be doing is human cells that are grown in the bottom of the dish. There are many of those cell cultures. Many people have given their tumor cells or other cells to scientists to grow. And there's been some controversy in there as well. Some people took the cells, some of the scientists took the cells without the permission of people. And, but right now there's no human embryos or anything like that being studied. The only place that human cells are involved is in the bottom of petri dishes growing under fluid. And those cells are passaged hundreds and thousands of times already. They were developed decades ago and have been passage.
Okay. Going back to COVID related, do you have any opinion about vitamin D as a preventative? Helen Antoniak asks.
There have been some folks that have said that that vitamin D might be good. Many older people don't get enough vitamin D to start with, okay? So I don't see any harm in taking a multivitamin every day. A lot of doctors recommend that to keep your vitamin D up. Whether mega doses of it, that hasn't been shown to be helpful. Whether it's being studied, there's a lot of work going on many different things. So if you're older and therefore at higher risk, and if your vitamin D might be low, as it is with many older people, then you could see where vitamin D supplementation might help you. But there's no definitive, take a whole bunch of vitamin D, a handful of it, and you'll get rid of COVID.
From Shannon, is there anything leaders, doctors, etc., can do to prevent this when the next disease does show itself?
Well, there's been a lot of attempts to develop vaccines that would be, or vaccine methods, like the messenger RNA method that we talked about earlier, that could develop vaccines quickly. So one approach was, if we're going to have an unknown attack from a new virus, let's be able to make a vaccine quicker than we used to. And that's actually happening now. We're making a vaccine very quick because of this method. We're also putting lots of vaccines. So that's sort of a generic way to protect against anything. You can look at things like influenza and say, that's highly likely to come back. We're going to have another, influenza just keeps changing all the time. We're going to have another Spanish flu sometime. And so you could start looking for ways to make influenza vaccines that are universal. Think back to the picture of COVID. Influenza looks a little bit like that. And those little lollipops that are sticking out of the soccer ball are what changed with influenza. But if you could make the soccer ball itself stays the same from year to year to year to year. If we could make a vaccine for the soccer ball, then we'd have a vaccine that would be against part of influenza that will be there no matter what the little lollipops change. So there's been a lot of effort going into a universal influenza vaccine. That's another approach that we're looking at. Making vaccines quicker, and also with influenza, trying to make a vaccine One other method that's being used is to take a whole bunch of influenzas, 8 different ones, and give them all to you at one time so that your body would develop a very broad influenza response. And hopefully if a new influenza came, you would have a broader response than you normally would. So those are the sorts of methods that we're looking at to try to develop a defense for any newly emerging disease. In addition, we're also trying to set up surveillances. There's a lot of money at NIH and other places going into setting up surveillance systems in China, in those open markets, with Chinese investigators collaborating, where they're constantly testing and looking for any disease that might pop out. So another approach is early warning system, trying to find it as early as you can. One lesson from HIV is if we had been faster with HIV and caught it in Africa before it got out of Africa, just think how much we would have gotten a head start by years and years and years and years. So looking and not being isolationist, but looking around the world, having partners around the world, working with places like the World Health Organization that I used to work with, and having an early detection system will benefit us in the long run as well.
Okay, Jim, we got a couple more questions. And we are running up to time. What is viral load?
Okay, that's how, in brief, that's how much virus you have in you. Okay, it's important. It's been important for HIV studies in the past. You don't want a high viral load. The more virus you have, the higher the viral load you have, the sicker you usually are. One of the ways that people are talking about it with coronavirus, interestingly enough, is how much of a load do you get when you get exposed? And there's been some interesting studies lately on masks. And it turns out that even if you get the disease when you're wearing a mask, the masks catch so much of it that you get a very, very, very small amount of virus when you get sick. And you end up with a small viral load, and you end up with a much less of a disease. So basically think of viral load as how much virus is in your system and how sick you are. The more virus, usually the sicker you are.
Okay. There was one on a mask. Hang on, Jim. I think this is from overseas. Oh, wait a minute. One more from overseas. What is the percentage of those who test positive become symptomatic?
Okay, one of the unique things about this virus is that so many people have it and don't show any symptoms. They walk around and shed the virus, but they don't have any symptoms. This is particularly true with children, but they're spreaders. They go home and kiss grandma, and grandma gets sick and dies. So this virus is somewhat unique in having a large number of asymptomatic people and makes the control of it much, much, much more difficult. There's been a lot of concern about they'll take people's temperature at the airport or in the doctor's office. And yes, if you're positive in temperature, you might have the disease. But if you're negative, you still might have the disease. So taking your temperature is one of those where if it's positive, it tells you something. If it's negative, it doesn't always tell you something. So the ratio is very high for COVID and depends a lot upon your age and your underlying condition. But remember, most people, as they get older and older, develop a lot of immune diseases. They develop arthritis. They're on immune suppressive drugs for their arthritis. That sets you up for a disease. So it's not unusual that older people have more disease and less asymptomatic.
Okay, Jim, there's two more quick questions. There's a slew of them, but two more because we're running over. One is, if I have used a paper mask one day, can I hang it up for more than 3 days and reuse?
Yeah, probably. Now they're recommending, that's one of the advantages of cloth masks. You can wash them. But basically, what you're asking is how long will a virus stay? There's been studies that look at how long viruses stay on surfaces. If you look carefully at those studies, there's an enormous amount of inactivation in the first 8 hours, hundreds of fold decrease. But some of the virus does creep out for many days. I would think that over 3 days, most of the virus in that mask, if you had any, would have dried out, and they don't like to be dry and then die. So you're probably okay. But they're relatively, now, thankfully, we're getting more and more and more masks. They're relatively cheap. And again, the cloth ones are washable. And here in New England, when it's getting cold, those cloth ones actually keep you a little bit warmer than the paper ones. So.
Okay, Jim, I have one last question, and then I'll turn it over to Deb for a conclusion. From JC, is there any data on blood types being more at risk?
No, there's a little bit of data that's just starting out, but not enough yet for us to definitively say that ABs are more sensitive than O. So there's some preliminary early data on that. It's unclear whether it'll be that simple that it'll just be blood type. Everybody's different genetically, and people, as we've seen, are going to process this infection differently. But early data suggests, that there is some differences. We'll have to get more data before it's definitive, but it will be interesting, very interesting to watch.
All right. Thank you very much for all the questions that came in.
Well, thank you. Many of them were really very, like I said, very perceptive.
So we appreciate the conversation with you, Jim, and thank you so much for your time and sharing your knowledge with us on such an important, subject, which is really affecting all of us in one way or another. Well, in many ways, I guess, to change our whole lifestyle as we've had to do. So thank you again, and thank you so much for everyone who came to the webinar, our first one of many, and we will give you a very big notice. I'll give you a lot of lead time on when the next webinar will be and what it will be for. But I want to also let everyone know, be aware of our tomorrow, Mater's Feast Day. We are going to be having a virtual gathering for a joint regional meeting with all 4 AASH regions participating together for the first time. And many of you who usually cannot travel to attend this, your regional meetings, you have the opportunity now. And it's from 4:30 to 6:30 Eastern Standard Time tomorrow. And just look on our AASH.org website, and it'll take you right to it. You can register, doesn't cost anything, and you can be a part of it. So once again, thank you, Rhonda. Thank you, Jim. Really appreciate all the information that you've given us on this subject. And thanks, and we'll see you again next time. Good night.